Welcome everybody. We have several people online, and we will go ahead and get started. This is the master thesis defense for Tamara Ali. The way this goes, for the sake of people like Dr. Isa here, is that the way we do this is we give Tamara 15 to 20 minutes to give us an overview of her thesis, and then we'll open it for questions from both internal and external examiners. Internal examiner here is Dr. Omar Dar Isa from the biology department, Bethlehem University. An external examiner is Dr. Isa Musa from the Environment Quality Authority. After they finish their questions, if anybody else has questions, and I may have also a couple of questions for Tamara, then we will basically meet the three of us, Dr. Isa, Dr. Omar, and me, make a decision, fill out the appropriate form, and tell Tamara if she needs to do some work or what specifically she has to do. This is how we normally operate. So with that, please.
Good evening, everyone. Today we are going to talk about molecular systematic of beetles from Palestine. I studied the evolution relationship among beetles by molecular techniques. In the beginning, beetles, ordered Coleoptera, are one of the most species-rich animals in the world. Unlike most insects, their front pair of wings can be hardened into wing cases called elytra. As the largest of all orders, Coleoptera, with more than 400,000 described species, also as a biodiversity indicator due to their habit specificity. Beetles' conservation is essential, considering their importance to the global biodiversity. Also, beetles play a vital role in the ecosystem, and they are important in decomposition and pollination and pest control. Without beetles, our environment would be overrun with the waste and debris. Meliridae and Dermistidae, both families of beetles that belong to the order Coleoptera. Meliridae is commonly known as a soft-winged flower beetles, while Dermistidae is known as a skin or hide beetles. These families share same similarity in appearance and behavior. One key difference between Meliridae and Dermistidae is their diet. Meliridae typically feed on the nectar and pollen, while Dermistidae are scavengers that feed on the dead animals and animals' products like feathers, hair, and skin. Both families play important role in the ecosystem by helping to decompose organic matter and pollinating plant. Quick background on Palestine. Palestine is one of the most biodiverse places on the earth, as we see on the photo on the right, because it's where their continents, Africa, Asia, Europe, and many different biogeographical zones meet. That has been shaped by its unique geological and climate history, also its distinctive topography and geological history. About 30% of the world living species can be found in the Palestine, and about 51,000 species are living in the historical Palestine. For animals, there is 30 and 815,000 species of Palestine, and beetles in the Palestine. Over 40 families of the beetles are known from Palestine. Below this is a table of the list of the family with selected reference from some of the most common ones found in the Palestine. They're little about molecular systematic. So molecular systematic is an available tool that is used in a molecular genetic to study the evolutionary relationship between different individuals and species, and has revolutionized our understanding of the tree of the life. This approach has provided important insight into the process that have driven to the diversification of the life on the Earth. Also, to provide the insight into a history of the groups of organisms and the evolutionary process that create diversity among species. By analyzing genomic data of organisms, researchers can reconstruct their evolutionary history and better understand how they are related to each other. Research objectives. So the molecular systematic study of the beetles in the Palestine, it's important because beetles are an important group to the study, and they are represented highly diverse and ecological. And they important group of insects. They play key role in the many ecosystem. And Palestine is a region with a complex geological and climate history, which has likely influenced the evolution of its beetles. With this knowledge, can it provide important insight into the evolution history of this region, and it informed conservation effort and better understand the ecological role of these insects, and to contribute the Global Gene Bank database by providing sequence of the Coleoptera of Mediterranean. So what we do, we collect specimen by using a set of trapping method. This figure shows this trapping method like in the beating sheet, insect nets, and hand collection method from different regions of a country. Was imaged all specimen, then selected through the image by InSelect software. Then it was extracted mitochondrial DNA from their tissue, then sequenced a specific region of the mitogenome, cytochrome oxidase subunit 1, CO1, which is commonly used as a molecular marker in insect systematic, and amplified DNA barcoding, and then sequenced by the Illumina. This figure shows an overview of a procedure. The laboratory of Dr. Alfred Vogerl has a specific methodology they do, and this figure summarizes this methodology that we use because based on the literature will establish methodology for DNA metabarcoding of a Coleoptera. That laboratory of Dr. Alfred is doing a global Coleoptera tree and phylogeny from a different country, and decision to add it Palestine. Then we align two families matrix data set of DNA sequence from Palestine with two global super matrix data set by Aliview. We use the phylogenetic analysis to reconstruct the evolution relationship among the different beetles species based on the DNA sequence. This involved the constructing maximum likelihood tree. We built it in the Mega. More than 1,500 insect specimens were collected from Palestine. Representatively individual specimens were selected for the DNA and barcoding, while the rest of the specimens were processed for the DNA metabarcoding. From the individual specimen, 22 Corculonidae and 7 Chrysomelidae and 80 Meliridae and 5 Dermistidae individual were successfully sequenced and DNA barcoded. These sequences were posted in the gene bank, and shared sequence were kept in the both museum, in Palestinian Museum of the Natural History and National History Museum of London. A taxonomic maximum likelihood tree was made that includes Palestine and parts of the country around the world to see the extent of the conversion between the origin of the Millardy and Dermistidae and other countries. Millardy molecular data, there are 80 Palestinian specimens where some identified to the species level. This figure showed the image of 80 Palestinian specimens, and this table shows the ID of these 8 specimens. Then compared it to 199 Millardy sequence from the rest of the world. Phylogenetic relationship of the Millardy of 199 specimens global that are available, and there are 27 branches here as we see in the tree. There is the molecular data. There are 5 Palestinian specimens analyzed. Some were identified to the species level, and this figure shows the image of all these 5 specimens, and this table shows the ID of this 5 specimens, also compared to the 86 Dermistida global sequence. The phylogenetic relationship of Dermistidae in ML3 of the 86 specimens global that are available, and there are 101 branches here as we see in the tree. A discussion over the past decades, mitochondrial genes and the genome have been frequently used in the insect study. There are several studies used a combination of a molecular and morphological data to reconstruct the evolutionary history of the beetles. In our study, we report the first molecular phylogenetic analysis, including Palestinian sample for the 2 Coleoptera families, Millardy and Dermistidae. Based on the analysis of our ML3 maximum likelihood tree of Millardy, it was found that malicious position within the superfamily Chloridia, and suggesting closeness of the malicious Millardy family to the superfamily Chloridia, and these malicious specimens were indeed closely also related to the specimen of the family Baphylidae from Malaysia and Panama, and suggesting closest of the malicious to the family Baphylidae, and the Millardy is the largest group of the superfamily Chlorida. Yanni 2021 suggests a new family including things like Malicious and Malicidae, which is separate from the rest of the Millardy in sensu lato in the wide sense. So our tree showed indeed agreement with Yanni tree. Also, based on the analysis of our Millardy tree, it was found that Diastae species were indeed closely related to the specimen of the superfamily Chloridia, suggest closest of the Diastena Millardy family to the superfamily Chlorida. Also, our tree showed indeed agreement with the young tree. These results are also the subgenus of Diastena virens is close into the phylogenetic tree to the Diastae aratus and Pampios obscures, and these results are construct with the previous study that have shown the close relationship between Diastae species based on the morphological and molecular data. Also, Yanni 2021 suggests the independent family of Diastena, which is separated from the rest of the Millardy and instead subfamily of the Millardy in sensu lato. Phylogenetic analysis support Malicidae and Diastae as an independent family instead of the subfamily of the Millardy sensu lato, and our results agree with the previous study. Additionally, it will be important to further the investigation, the evolutionary connection of the Milleridae family grouping or Coloridia. In Dermicidae tree, the phylogenetic of Dermicidae has made progresses in recent years using mitochondrial data, and based on the analysis of our Milleridae our Dermicidae tree, it was found that Anathrinos, the sister group of the Tragodorma, and all the smaller genera of the Anthrinos, which is constant with the result with Zhang in 2021. The phylogenetic analysis found that the Tragoderma and Anthrinos are each monophyletic. So our results agree with the previous study. Phylogenetic biographical studies have been used and investigate the historical biography of the petals. Also, they can be used in infer the historical pattern of the dispersal and the variability in petals and understand the diversification and distribution of organisms over the time. Our result in the Dermicidae tree shown one specimen of Palestine in Palearctic region branched next to the specimen of the same genus from the South Africa Afrotropic region and the Canada in the Nearctic region. Also, in a Milleridae tree show one specimen of Palestine in the Palearctic region branched to the next to the Malaysia, Indo-Mali, and Panama Neurotropic. There are geographical separation because Palestine has a very rich insect variety as a result of its geographical location and geological past, making it a part of the Eastern Mediterranean biodiversity hotspot. In our study, we used the cytochrome oxidase as a DNA barcode marker for a molecular identification and delimitation of the species in the beetle and other insects, due to the high mutation rate and conserved region that allow for the design universal primer. However, the use of the cytochrome oxidase as a single marker has a limitation, include potential incomplete ligand sorting and hybridization and integration, and which can lead to misidentification and incorrect species delineation. So must use additional marker like other molecular mitochondrial genes or a nuclear marker. It's may necessary to confirm. The use of the mitogenome sequence in a molecular taxonomy can provide more comprehensive understanding of the evolution relationship among taxa. However, sequencing and analyzing the intergenome can be more costly and time-consuming. In conclusion, our study reports on the first DNA barcodes petals from Palestine, which is reconstruct the phylogeny of Milleridae and Dermicidae based on molecular and morphological data. And phylogenetic analysis are an important tool of the understanding the evolutionary relationship among species. Our recommendation for further studies include additionally markers and morphological character are needed to understand the evolution relationship among genera and species, reassessing taxonomic nomenclature, also apply the similarity methodology to other families of Coleoptera in Palestine. Publishing aim to separate them into 3 sessions, Dermicidae, Malicidae, and Dermicidae, ensuring molecular data and names align with the early taxonomic nomenclature and morphology. Also explore molecular studies utility in ecological and conservation study, particularly in addressing threats like habitat loss and pollution and exploitation, invasive species, climate change, Israeli colonization. Finally, exploring whole mitochondrial genome, rather the cytochrome oxidase barcode region can produce more robust phylogenetic trees. I would like to thank all those who helped me, Professor Alfred Vogler and Professor Mazen Komsuya and Dr. Hushi and Mr. Muhammad Abu Sarhan والدكتور عيسى البرادعية والدكتور عمر دار عيسى. Thank you.
Thank you. يعطيك العافية. Okay, so we open it now for questions. Maybe I'll leave the projector on. We open it for questions first from the internal examiner, Omar Dar Isa. Dr. Omar, if you have any questions.
Yes.
And then we'll take some questions from Dr. Isa, and we can come back to you in the second round of questioning. But anyways, please go ahead, Dr. Omar.
أول شيء يعطيك العافية تمارا. Okay.
زيدك دكتور عمر.
First of all, I'll start with the technical issues. I left you many notes in the thesis. I will send again. You have to put the format according to the MBT program of Bethlehem University. Okay, some paragraphs need to be rephrased in your own language. Behind this, I will start with your abstract. I quote, you said, we conclude that morphological differences do not predict genetic differences in beetles. This is based on your study on the COX-1 gene. Does this mean that the classification of beetles based on morphology in the past has changed according to your new classification or phylogenetic clustering based on the COX? يعني في اختلاف بين تصنيف السبيسز اللي انت درستيهن وبين النتائج اللي طلعت معك. طلع يعني انه هذا مش لازم يكون هيك، هذا لازم يكون هيك ولا
دكتور انت مش فاهم كيف انه هم بيسعوا انه ما تكونش in instead بدل in family subfamily in Miliridae انه تصير instead to the family لحالها not under the Miliridae like the Diacity and Malicious in the sense Lotto.
So I didn't get it. What does it mean? The morphological differences do not predict genetic differences based on one gene. Based on one gene, الكوكس يعني. This is a conclusion of your own work. يعني بالabstract. You know, maybe the COX gene has no phenotype that you can see by eyes. So if you want to read into such a conclusion, you need to do what you have suggested in the recommendation to study, you know, much more genes.
Oh, okay.
Okay. Okay. Another thing, Tamara, in the literature review, section 2.2, the last two paragraphs, there is a reference you used from 1913 for Zalberg. It's talking about this is the first description of beetles in Israel and Palestine. And I wonder if there were Israel in 1913. What was that a typo or what was the problem with that?
واحد.
الدكتور بجوز هو الريفرنس كان مكتوب بإسرائيل وهم ما طلعوا بإسرائيل عشان كتب فلسطين جامعي.
لا نغيره.
لازم نغيره؟
طبعا.
But there was no Israel by that time. Okay. نعم دكتور مازن في شيء؟
عمر، بس تقول بالنسبة الأولى، النقطة الأولى اللي حكيتها regarding the first point, I also agree with you. We have to change the abstract in that sense. Actually, what she meant to say probably is that the taxonomic literature is not consistent with the molecular literature, not the morphological differences.
That's true. طيب. Another question, Tamara. There are several methods to do the calculation for the reconstruction of a phylogenetic tree, and you choose the maximum likelihood. You could also use the parsimony, maximum or minimum parsimony.
Okay.
My question is a general one. Why didn't you use, for example, the joining neighbor or obgema methods? Of course, you choose the correct one. Maximum likelihood is good for your case. But why not the joining, neighbor joining method? Do you know the difference between them, the ML and the NG?
Of course, maximum likelihood, it's في bootstrapping between the branches يكون close أكتر من النيبر joining. bootstrap أكتر.
Neighbor joining عادة we use it normally not for sequences, for bands, like for microsatellites or something that you did by banding profiles.
And our boundary tree of from the global, it's maximum likelihood also. So
for
we
sequences.
global sequence.
Do you know the major drawback? One drawback. Give me the main drawback of the ML in general, the maximum likelihood. Other than it consumes too much time to calculate.
Maximum likelihood is have more advantage than the neighbor joining.
Of course. But do you know the major problem with the maximum likelihood? Of course, all of these are prediction and calculation, and they never give you 100%.
Maybe consuming time when analysis. It's
of course, other than that, I tell you, sometimes it failed to take into account many factors of sequence evolution, like the convergence and homoplasy. For example, insects and birds, they have similar organs doing similar function, but they have no common ancestry in terms of the genes.
Okay.
Even with the sequences, this is the major problem with the maximum likelihood. But anyway, so some other thing. You said in your presentation and in the thesis that you deposit the sequences in the gene bank, but also you kept, I don't know what you kept exactly, the sequence or the samples themselves, the DNA, in the museum. The two of them.
Because we take it as the mitogenome from the tissue without broken the insect body.
The DNA or the tissue?
Without just from the out tissue. Without يعني نضر العينة يعني.
Okay. How did you get them? كيف أحفظيهم؟
كيف حفظناها؟ حطيناهم بإيثانول فرزناهم.
Okay. So there is also one comment. You know, the phylogenetic trees that you produced are too big, and you tried to trim them to be able to present them in the thesis. And the trimming, you know, I didn't see the out groups, and in some trees, like the one you presented in your presentation, there is no label for the Palestinian samples, and it's hard to see. My question,
في label
in the circular one.
في label لونهم أصفر البالستيني.
No, فيش label في المدورة. الكبيرة هاي أكبر واحدة
ندور
اللي
على أصلا
related.
تتقابل.
Anyway, my question, if you try to take your sequences for the Palestinian specimens with those closely related sequences, the international sequences, and try to reconstruct a smaller tree, do you expect to have the same clustering pattern if you reduce the number of international sequences only to those who are around the cluster where your Palestinian samples are, and to get a more, you know, a more clear tree?
دكتور ممكن تعيد السؤال لأنه البرزنتيشن راح اللي نسمع نسويه.
بسيطة بحكي لك. انت جبتي العينات تبعتك وحطيتيهن السيكونسز مع 199 واحد اجنبي من العالم.
اوكي.
اذا اجيتي على على العينات العالميه اللي قريبه من اللي الك من الفلسطيني،
تمام.
يعني وتختصري تسوي الشجره للثيسز مش للبروجكت. I know the project is international and you need all the sequences. هل راح يطلع معك شجره phylogenetic tree تشبه الأصلية اللي عرضتيها وفي هيك حالة بتقدري تعرضيها بشكل أوضح، ولا بتتوقع يتغير الكلاسترنج بروفايل when you reduce the number of sequences according to the maximum likelihood؟
هلا السيكونس تاعتنا بدنا نحطهم مع جلوبال كمان دور؟
بس مش كل جلوبال، تختاري بس السيكونسز اللي في نفس البرانشز حواليهم يعني.
لا أكيد راح يتغير.
طب جربي، إذا في اوت جروب المفروض إنه يكون ستيبل. ما أنت بتعملي ريدكشن بس مش يعني لكل شيء.
لا بتغير، لا جربنا بتغير لأنه البرانشز والتاكسا بصير ترتيبهم أوسع للتري، فممكن تشوف إنه صار عندنا التري أصغر، صارت الماكسيموم لايكليهود والبوت سترابينج بيشتغل أحسن لما نقلل عينات كمان، لما نختار بس هاي السيكونس.
يعني موقع البالستينيان بيتلز راح يتغير؟
لا بس إنه ممكن العينات اللي حواليها تتغير، يعني بس مش كثير، يعني ممكن بس إنها تبعد بس تاكسة واحدة، تاكسة from 2 branch 3 branch just.
اوكي يعني you tried this already؟ جربتي هذا الحكي وتغير؟
جربنا، احنا عملنا اكثر من شجرة لحتى وصلنا هاي الشجرة.
اوكي.
وكل مرة كنا نقارن البرانشز.
طيب بدي اسالك كمان سؤال. بتعرفي in Palestine we have several climate zones as you said, these contribute to the huge biodiversity we have among the plants and animals. My question, the beetles that you studied, which zone is best or they like more to live in? When we have the desert, the Jordan Valley, the Mediterranean, the coastal area.
As I said before, beetles is different. Like Meliridae, it's founded in the plants because it's important to the pollinating plants. So it's not in the desert, like in climate good, not dry. But Dermestidae, it's founded in the dead animals and animal products. So we found it everywhere and not in the plant, just in the, like scavengers.
Yeah, I know. You said that. But my point is, other than the host,
found it in the agriculture. It's important in the agriculture. So we found it in the area with the plants with a good climate. But the Dermestidae, it's founded in
Okay, اسمعني. Jordan Valley, we have plants, but the temperature is much higher than the Mediterranean area. We have also plants. In terms of the rainfall, the temperature, the soil type, are both, whenever we found plants, we found Merilidae, for example, the same species? or different species, or
Of course, we can find different species from different region in the world, because Palestine is the hotspot biodiversity global. So we can find, like in the map, we have geographical separation, and Palestine, its climate, it's a change over the history. So the beetles, when beetles believe in the past, cannot live in this time, because the climate is a change and the distribution of it is change also.
And my worry was about if certain
This
species
show zones.
اسمعيني. if certain species like a certain climate zone, okay, then the diversity rate will be slow among time, because they all under the same selection pressure. So in this case, regardless if we have 3, 4, 5 different climate zones in Palestine, they are only found here. So the diversity in the climate and the temperature and whatever has nothing to do with the diversity of these species, because they are restricted to one zone.
No.
And this leads us to your trials of correlating the geographical distribution of the beetles globally, because you know some samples found here in Spain, in Africa, in different countries with different climatic zones, and it's hard to correlate from these results if there is any relation with the geographical distribution with these results, the phylogenetic clusterings. I think we need much more into
of course,
this.
we need. This is a first study that studied the phylogenetic with biographical in Palestine. This is a first study, so we cannot sure what is the exactly zone Millerdi and Smith live in it.
But as a conclusion, in one sentence, as a conclusion, could you say for sure that these results can establish a correlation according to the geographical distribution among the different beetle species globally that you studied?
شو؟
يعني ممكن نطلع في conclusion ما بين الفايلوجينيتيك كلاسترنج اللي عملتيه التري and the geographical distribution for sure.
اكيد دكتور، هدول بس جست مثال انه درميستيدي، انه هاي بس سبيسيمن واحده، في other specimen ما طلعتش يعني في البالاريتيك region، يعني but just is a sample from the large tree، انه there are a separate geographic and we get reason because Palestine is a global biodiversity فيها it's diversity فيها عالي, high rich diversity in the insect especially.
Okay.
يعني so, in the tree we have some specimen. It's good. It's in the Europe, in the like same Palearctic region, but some other is no, because like I said before, Palestine is
who, by the way, okay. Of course, we need some other, maybe also morphological constructing to have such correlation. But anyway, one last thing before I go to Dr. Isa, طولت عليكي. The sequencing using NGS.
Okay.
Where was this done? وين هذا العمل؟
In London.
هو ومين عمله؟
دكتور هوشي.
اوكي إذا دكتور عيسى يتفضل.
طيب يعني هنفتح المجال الدكتور عيسى will ask a couple of questions. Also, Dr. Alfred, after Dr. Isa, I'd like you to comment on some of what you heard and maybe answer some other questions, ask some other questions. تفضل دكتور عيسى.
That comes from the Environmental Quality Authority, and we are an institution just promote the environmental sustainability, and we care about biodiversity conservation. And such work give us an insight just for the future. I mean, just it's a valuable information that you have just presented in your thesis and your work. So it's highly appreciated. And we just feel that it is an original work has been done in Palestine, which we need just to build on it as an environmental quality authority for our, I mean, strategies for our work, for our decision-oriented, I mean, just that will help the government to put a plan for conservation. And this is also, it's a part of our conservation strategy, which we have done recently. So it is very important to us. I have seen that in your presentation and in your work that 1,500 specimens have just been collected from Palestine. And you have done, this is the DNA analysis and barcoding and sequencing for some individual. 42 individuals have been successfully, I mean, just barcoded. And some of them has not been successfully, I mean, just given a result. So what was the reason, I mean, for that unsuccessful result for some of the specimens?
Because we have a problem in the transport to the London transport process to London, because it's late to get it and put it in the fridge.
This was the reason, because a few specimens had been successful, only 42 of
No, there are more than 40. There are 120 maybe, but not it's coleoptera just. There are other insects. And the other specimen, it's go in the process with the meta barcoding, not the DNA barcoding. Meta barcoding, because it's not coleoptera.
في بعض الملاحظات بعد في على الثيسز أنا العلاقة بالرايتنج. لازم
شفتها.
تاخديها بعين الاعتبار يعني.
عدلتهم.
شكرا
على ملاحظتك.
طبعا هذا المشروع هو طبعا مشروع كبير بسموه لونج تيرم بروجكت ريسيرش، بدأ في سنة 2018. أكيد في نتائج كثيرة جدا. يعني أنا بالذات التحليلات اللي لها علاقة بالدي ان اي هي not reflected in this thesis، وأنا بعتقد إن ما تم وضعه في هذه الثيسس هو كافي لرسالة ماجستير، يعني كافية. ورسالة أنا طلعت على، يعني كنت عضو في أكثر من لجان طلبة ماجستير في الجامعات الفلسطينية. للأمانة ما تم تقديمه من خلال هذا البحث، والبحث اللي قدمته زميلتي السابقة اللي هي أمل حنظل، يعني مستوى يعني أعلى من مستوى ماجستير في جامعاتنا الفلسطينية اللي احنا تعودنا على يعني العمل تبعها. فطبعا في أنا عندي ملاحظة إن احنا لما احنا قارنا الجينيتيك ماتيريال ما بين عدة أصناف في دول العالم أخرى، ممكن في المستقبل أن نروح باتجاه مقارنة البيئات. يعني البيئات مثلا زي ما حكى الدكتور عمر إن احنا في عندنا 4 أو 5 أقاليم مناخية، نقارن الأقاليم المناخية للأصناف الموجودة عندنا مع الأقاليم المناخية الموجودة في الدول الأخرى، ونشوف إيش تأثيرها على الجينيتيك ماتيريال. أنا بكتفي بهذا.
شكرا لك كثير ملاحظتك القيمة.
دكتور مازن، ممكن بس ملاحظة صغيرة، بعرفش هاي لفتت نظري. في الانترودكشن في فلسطين في 373 سبيسيز اوف بيردز، 373 نوع طيور تحكي على التبع في فلسطين. أنا اللي بعرفه من أكثر من مصادر إسرائيلية ومصادر أريج، إنه في 511 ما بين مستوطنة وزائرة وشتوية وصيفية وفاجرانت وكله على بعضه. 500 في هيستوريكال بالستاين في 511.
يعني نعم، يعني في اختلاف حوالين الأرقام، بس احنا راح ناخذ برجع للرقم بتأكد إنه بتابع مع الان بي ساب والاي كيو اي يعني.
هلا المستوطنة 282، نعم المستوطنة، أما كلها على بعض 511 حسب ما بعرف.
الأعداد الموجودة عندنا في الضفة الغربية قطاع غزة 300 زي ما هو مكتوب 374 وزادت واحد 75، لأن ما هو موجود في فلسطين التاريخية كما قلت أنت بتوصل 540 تقريبا 42.
ما هي كاتبة إن هيستوريكال بالستاين فيها القضية بتغير إن ويست بانك أند غزة، يا بتغير الرقم.
Okay, thank you. We'll conclude for the sake of Summarizing for you the last 2 points because they were made in Arabic. Dr. Isa mentioned the importance of such work for the future, for the work of the Environment Quality Authority. Dr. Isa represents a natural resource section of the Environment Quality Authority. We collaborated with him on a number of projects, including the Protected Area Network and the National Biodiversity Strategy and Action Plan. But he explained that this is better than other master theses he has seen in Palestine, but that there's a lot more work to be done in this area and would be beneficial for conservation efforts. There was a little disagreement on the number of species of birds, but that has to do with we have to be consistent. And Dr. Isa thinks there's 374, 75 species in the West Bank and Gaza. If we take historic Palestine or Palestine in general, it's probably more than 500 species. Anyways, I open the door for you, Dr. Alfred, and then we'll come back to the two examiners. Feel free to ask tough questions of Tamara if you like. Put her on the spot.
Well, that's not my intention, but I mean, I think there is a point. So if we use like Coleoptera as one of these indicator groups, right, that we want to build the conservation strategy around, so I think we need to get it right. So I think it needs to be well argued, and I thought, I mean, in particular from a discussion with Dr. Omar that you had, I think there was a bit of a lack of sharpness there, and so that's what I would like to ask you a few questions along these same lines. And I don't know how much time do you have?
Your voice is gone, Dr.
Yes,
Alfred.
I'm here.
I don't know why.
Please go ahead. I can hear you.
You can hear. Okay.
Yeah.
All right. Tamara, can you just present your...
Just 1 minute, Alfred. Let me restart this.
Oh, it's closed. It's not critical. I just wanted to make one point, which we may remember. No, if it's...
just 1 minute, Alfred.
Don't bother. I try to describe the figure.
Give us 1 minute. Technical issue.
Oh, I
Just
see.
1 minute.
كلنا وانت سامعنا.
خليه يجي ولا بطفي كمان. لا هو بيجي. خلاص نشيله ونسمع من هنا.
خلاص اشيله من هنا.
على الصوت ماكسيموم. ايوه. Go ahead, Alfred. Repeat the question.
Tamara, you had one figure, and I wish we could see this now, but so you had like Palestine and all kind of arrows going all over. Can you, just because we cannot see it anymore, can you just describe how the figure looked like?
Can you just describe what the figure looks like, the one with Palestine?
الخارطة اللي فيها طريق هجرة الطيور.
ادخل على النت واجيبها يعني؟
لا احكي عنها. انت ورجيتي خارطة.
كلك
شو الخارطة بتورجي؟
إذا بدكم بعمل شير أنا الخارطة.
هي.
Are you sharing your screen?
شير معمول.
Go ahead, just explain.
Yeah, so that's a little further. There was one.
Yes, figure.
No, like with arrows coming from.
لا الخارطة اللي فيها الخطوط الهجرة الزرقاء في المقدمة.
اوكي. خارطة فلسطين مش خارطة العالم.
ايوه.
في خارطة العالم.
ارجعي.
طالع فلسطين. هاي.
لا ارجعي كمان مش مبين عنا. ايوه.
That one. Very good. Yes. So do you think this is a useful figure to describe what you want to research?
That's a very good point.
Maybe because Palestine is there between three continents, Asia and Europe and Africa, just to mention.
Well, I know why you mentioned it, but is it a good figure, a good graphic idea of what we are trying to work out?
Maybe to see how Palestine is like a hotspot to biodiversity with the three zones.
Okay, so what do the arrows mean?
Just to share to Palestine position on the world map. I don't take what mean exactly, just in Palestine, and they're in three continents, Asia and Europe, and
Dr.
just
Alfred,
not to
may I
mean.
interfere, Dr. Alfred?
Sure.
Yeah, this is
This
the
is
migration routes of
I
birds across
this very
Africa to
information.
Europe and the other way around. And about 60% of birds
بعد الطيور
take
بس أنا
Palestine as the
mention.
resting place during their migration. So
So
it
that's
has
from
nothing to
birds.
do with beetles, but it has to do that birds prefer Palestine through their migration to Africa or to Europe.
Thank you for this. Yeah. So, okay. So then the question remains, so what do you actually want to test? So, okay, this is taken from birds, and I appreciate birds travel long distance, overwinter somewhere else, etc. So it's just a kind of a transition route. But what do you think about the beetles? They don't do this, right? They don't overwinter in southern Africa, for
Yes.
example.
No.
So what is it? How would that? What's your model when you talk about the biogeography and the phylogenic history of the beetles in Palestine?
That's a very good question يا فريد. ترجم لك بالعربي، يعني شو فايدة هذا الحكي اللي بتحكي، بتقول إنه بين الكونتيننتس. أوكي ماشي، شو فايدة هذا الحكي بالنسبة للبيتلز؟ شو علاقة الموضوع هذا بالبيتلز؟
It's important for know the historical evolution for this region, exactly. Historical evolution of this region and
Okay, and in terms of arrows,
tell us how the historical is it's come in the future, maybe in the past it's different from the
right?
no.
So do you think of Palestine as an area of like ancient diversity, or is it an area where things have migrated into?
فهمت السؤال؟ I'll translate the question. فكر إنه هي المنطقة، يعني شو هل البيتلز هذول اجوا من برا يعني هاجروا إلها، ولا كيف تأثير هذا الموضوع، أو شو اللي بيخلي فيه تنوع حيوي للبيتلز بفلسطين؟
Because
كيف شو علاقة المواضيع هاي، موقع فلسطين الجغرافي والجيولوجي والشغلات هاي مع التنوع الحيوي؟
Because Palestine region is there have many zone meets, so we can found different beetles from many zones of the different these three continents. So these beetles come with by use Palestine as a road to come to the Africa, like. So the beetles come from different region,
Right.
like in the some of the foods we get it المسافر المحمي، ممكن beetles تيجي من كل مكان.
Okay.
Dr. Alfred.
Yes.
I believe that the ancient biodiversity in Palestine was much more than today. We used to have thousands of years ago woodland, and we used to export some wood and other stuff to the neighboring countries. And many nations come to this area, and they bring their animals with them, maybe beetles along by mistake or by chance. And because of the so many factors that we have today, from the colonization, from the separation, the segregation wall, from the climate change, from the use of land for, urbanization. So we have habitat loss, habitat fragmentation, we have hunting as part of the Palestinian culture. So we are expecting reduction in the biodiversity in all the species, not only the beetles.
Oh yeah, of course.
I may just go back to the original question because I think it was not answered properly. The question can be rephrased. For example, why do we have in such a small country called Palestine, historic Palestine, why do we have 4 biogeographical zones, each with its own distinct flora and distinct fauna, including of beetles? Why? This is the historical thing that Alfred was going after. What's happening here? What's happening here? We're actually working on a paper along this line of biogeography, Alfred, which might be, I'd love for you to be a collaborative paper, because we talked about where and when flora and fauna penetrate into Palestine after the recession of the Tethys Sea and the last glacial age and all of this. And then when did the Ethiopian Sudanese elements come here? They came along the Rift Valley. That's why when you went with us to the Jordan Valley, we found some interesting species there. That comes from the Rift Valley geographically and biogeographically. And then third, the desertification, which is what Omar is alluding to, that we have climate change and other things. We have increased desertification, so we get Sahara-Arabian elements now increasing in their representation, including of beetles. I think this is worth investigating, not just for beetles, but for plants and for everything else.
Yes.
Well, but I think, it's Tamara's defense here. So it's like, I think I need to, drill that down.
No, I'm grateful for putting her on the spot.
I'm not putting on the spot, but I think I want to clarify this also,
I want
that
to
in
put her on the
everyone's
spot.
mind here. So what does the tree really tell you in this regard? So, okay, we find like in Palestine, so there are these, let's say, Sahara African elements there. So we need to go through the exact details you said. So in the Dermestidae, they are closely linked to the Afrotropical and Oriental groups. So can you just be more precise? What is it when you look at the phylogeny tree? How have you established these facts? So the fact that the Palestinian groups are derived or that are linked to the Afrotropical and the Oriental lineages. How do you use the tree is a broader question.
The tree show us Palestine come in what region close to it. Palestine is a correct come Palestine in the Palearctic region in the Europe and Asia, but Africa and Indo-Mali is far than a Palearctic, so most the specimen of Palestine show in the Palearctic region, like Germany, Spain is a Palearctic region, but the tree show some specimen is have a separation in geographical map because Palestine is a hotspot in 3 countries and 3 zones meet. So
see, I think you're starting with the conclusion and then fit the data. It needs to be the other way around. So you were just saying that some of the closest relatives are actually in whatever you said, in Germany or somewhere in the Palearctic.
Paleolithic, yeah.
Is that right?
Yeah.
So what does that mean? Or what does it tell you about the Palestinian fauna and its importance for conservation?
Because it's important in the conservation to tell us the how to relate it to each other and the historical evolution between these species is tell us this is important to conversation and to understand the relationship between it.
Well, but you could argue, well, if their
Important
opponents,
in the ecological study, it's important like in agriculture and pollinating.
hang on,
And
that isn't my question. The question is just about the tree. So you just said that the closest relative is in Germany, let's say, or somewhere in the Palearctic.
yeah,
So could that not mean, well, if it's all over, why bother with Palestine? Why make that big case?
Yes.
not big,
Maybe Mazen translate, maybe.
not
No,
a big
you get
problem because it's
أنت
connected in the
ترجمها يا عمر.
Palearctic region.
السؤال.
Is it allowed that I translate
Yes,
according
go ahead.
هو بحكي لك اذا فهمت صح انه السبيسيس الفلسطيني طلع قريب على واحد في المانيا في شجرتك. كيف بتفسري هذا الحكي؟ هذا شيء كبير، وين المانيا وين فلسطين؟
That is, it's a Palearctic region. It's correct. Get it Spain and Germany to close to the Palestinian branch. So it's a Palearctic region, and this Palearctic region's zone may be different from Africa and in the Mali. So it's the beetles from the Palearctic in Almania and Palestine have the same zone to live in Palestine and in Germany.
Well, in that case, you could argue, well, if we lose a little bit of that bigger area, i.e. Palestine, it wouldn't matter, right? So I think my point is really you need to look at, I think, different levels, different hierarchical levels. So if you want to make the case that Dasytus plumbeus or so, which is a very widespread species, that it matters in Palestine. Actually, all of the ones that you picked are rather quite widespread species, right? So that they themselves have very little, I mean, just as Linnaean name, they have very little conservation relevance. But you could maybe investigate if there are certain genotypes. So if there any evidence of geographic isolation in Palestine, right? So this is on that tip level where you look at. And then you also can look deeper in the tree. And there you can see these relationship of the historic geography at a much larger time scale. And you would then see, well, how are they derived? And I think it will be almost inevitable that very few of your lineages in Palestine are actually the ancestral lineages. So they're all in some way like an offshoot of an deeply Oriental group or a deeply Afrotropical group. It's just that they come together. So that's why I didn't like the bird example, because it just flips the whole thing around. So what your expectation is actually exactly the opposite. It's just like the crossroad of things. And it's very important to know, well, how distant are they from things that are elsewhere? And you can look at that in the widespread species, which almost in that exclusively you have. That's more a population level question. But you can also look at these maybe by taking some of the other lineages that we can discuss later. Look at the other lineages and see where you have like rare species or some other lineages that are not linked to humans quite the same way.
Yes.
And look at the deep historical origin of these, right? So that's something to discuss maybe later on for your paper, etc. So it's maybe too much for now. But I mean, the trees, I think it's very important. It's important to understand what the trees mean and how to read them.
Yes.
Yeah, of course.
This is, of course, if we assume that the phylogenetic tree based on the cox, a small part of the cox is, stable enough to reach into such conclusion.
Yeah, I heard what you were saying, yes. But I mean, this is, though we are doing it mainly on the basis of mitochondrial genome, so they
Yes.
are a little stronger. And so if we sample the tree densely enough, which we're trying, and there's still room for improvement, but they are not that wrong, I would say. Not that
Alfred, one aspect that's really important for us that also helps answer some of the questions you raised is that unfortunately Tamara doesn't have the location of these specimens. She's still not able to access files in a way to pull out where they were collected in Palestine. And I'll tell you why this is important. I mean, Anthrenus as genus is found throughout Europe and throughout the Middle East and extending into Asia. But the branch of it that's Palearctic is very different than the branch that's Oriental, if you want.
Yeah.
We don't here have representation, of course, of all the species of Anthrenus or of Trochoderma. These are very few specimens and very few taxa. So you are absolutely right. You need to read the tree cautiously, and you cannot say just because one specimen from Palestine showed up to next to one specimen in Germany, that means that Palestine is somehow closer to Germany, either geographically or in any other sense that we can think of. And that conclusion, you have to be very careful in the way you interpret this data. And this is, I think, your point about the trees and taking them not as trying to explain your pre-arrived conclusion that we are part of the Palearctic, which is in this figure. We're really not part of the Palearctic. We are part of, we have Irano-Turanian elements, we have Afrotropical elements in Palestine, and we have Palearctic, and we have also the Saharo-Arabian elements. But we don't have the data on this yet. So we don't have the answer that you are seeking.
So yeah, I mean, if it's just a matter of us needing to supply the data, one thing, and I think we need to wait a little because, as you know, we have that second set of specimens that we collected when I was there. I mean, that is still being processed, right? So, but very soon, I hope we get these. And then we have many more families, many more lineages that we can test, and then our databases should be more accessible also. So that's yeah, I'm sure we make a lot of progress in that regard.
Thank you. Yes, absolutely. Do you have any other comments, Alfred?
No, I think that's. I think I should let you go. And it seems like there are. This is not really part of the thesis, so that's more like the further plans, which I'm very keen on to continue.
Okay, I have a couple of questions for Tamara. I don't know if you want to wait and hear my questions or you want to leave.
I mean, it's probably. A little under time pressure, so I stay on for 5 minutes.
Okay, so my first question to Tamara, I asked it also to Amal a couple of days ago, which is to tell me what, but instead of species, I want to tell me about genera, like this genus. What is your concept of genus and how do you understand the genus? Since you were talking about some things from the same genus, etc. What is a genus? Can you define for me a genus? شو تعريف الجنس هذا الجينس Trochoderma genus and
come under the family genus order than family than genus. So
okay. So what is it?
It's
I know it's under a family and I know it's above species. That's what is it?
have the same ancestor to the family Meliridae, like Diacity. It's a offspring of the Meliridae, so we can adjust this is under this family by in this genus because it's the same ancestor with the family.
So how do you know that this, for example, here, Anthrenus, you have Anthrenus here and Anthrenus here.
Okay.
What defines that they are both Anthrenus? Why not give them two separate general names?
Because
defines
have
the
a
genus?
different sequence, so get every individual in this Anthrenus genera because it's had different, like two brothers have the same ancestor, so different genera.
Okay. My second question, we'll come back to that later maybe, but my second question is, what do you want to do next? You mentioned some recommendations for research. Do you want to do this research yourself, or do you see somebody else doing this research, or what is it that you want to do going forward?
Of course, we want to separate them in the three paper, like we said before.
Okay, but that's publishing what we have, and as Alfred said, we may have to wait till we have more data and better clarity on some of this to publish.
Of course, we want
But my question is not about this current data. My question is about this other recommendations that you mentioned, and who's going to do this work. من يعلق الجرس زي ما بيقولوا. Who's going to do this? Are you interested in doing research on this?
I want to complete in this and finish it to make it done to understand everything in these trees and in these families exactly, and give the historical evolution of these genera to. It's important to know in Palestine too.
Now, Dr. Isa, Dr. Umar, and any of the audience members, if you have any questions, feel free, including family members. اسألوها. Feel free to ask questions. Muhammad, do you have any questions for her? No? Dr. Isa? Dr. Umar? Dr. Umar معنا?
Yes. Enough. I had enough questions.
Okay. All right. So now I would like everybody, like we did with Amal, I would like everybody to kind of leave. Those online also can log out.